Let us discuss KCSE 2024 Biology Paper 2. On to our first question. The following word equation represents a biological process that occurs in green plants. Okay, so that means that this process is specific to green plants. Now, in our word equation, we have carbon 4 oxide reacting with water, leading to the formation of glucose and a product labeled E. Now, of course, glucose is a food substance. So essentially, we are having CO2 and water reacting, leading to the formation of food. So this makes this process photosynthesis. Part A. Name the cell organelle where the illustrated process takes place in green plants. Now, the organelle in which photosynthesis occurs in is the chloroplast. B. Roman 1. Give the likely identity of product E. Okay, so product E is going to be oxygen gas. Now, the reason is because oxygen is given off as a byproduct of photosynthesis. Roman 2 suggests ways in which the rate of production of E can be increased. Okay, so the question wants to know how we can increase production of oxygen gas. Now, production of oxygen gas is going to be tied to the rate of photosynthesis. So essentially, the question is just asking us for the conditions that need to be present in order to speed up the rate of photosynthesis. And these are as follows: optimum temperature, optimum light intensity, increasing the concentration of carbon 4 oxide. Part C suggests one possible source of carbon 4 oxide if the above process was artificially conducted in a school lab. Okay, now if this was to be conducted in a lab and we needed a source of carbon 4 oxide, the most common one would be sodium hydrogen carbonate. So sodium hydrogen carbonate will break down to form sodium carbonate, water, and of course what we want, carbon 4 oxide. Part D. Explain how the illustrated process is significant in nature. Now, the question wants us to identify the importance of photosynthesis. Now, photosynthesis plays a huge role in nature. Now, it does so through several ways. Number one is purification of air. So how does it do so? Now, when it comes to photosynthesis uses up carbon 4 oxide, right? So by using up carbon 4 oxide, what happens is that it reduces the concentration of carbon 4 oxide in the atmosphere, which is a good thing because carbon 4 oxide is one of the leading gases in global warming. So if we have less of carbon 4 oxide in the atmosphere, that is good. So photosynthesis plays a role by reducing the concentration of carbon 4 oxide, and by doing so, it purifies the air. Number two is that it also increases the concentration of oxygen in the atmosphere. So as stated, oxygen gas is a byproduct of photosynthesis. So when photosynthesis happens, oxygen gas is released in the atmosphere. And this is very good because organisms rely on oxygen in order to carry out respiration, production of energy. Okay, now last one is it provides foods. Okay, now a lot of the food substances that we obtain from plants are actually produced through photosynthesis. So we have photosynthesis to thank for all of these amazing contributions. Question number 2. In an experiment, the number of red blood cells present in 3 samples of blood labeled F, G, and H was estimated. Different concentrations of salt solution were then separately added to the samples. After an hour, the number of red blood cells present in each sample was counted and recorded as shown in the table below. Okay, now I am going to take my time with this question, okay? Because this question deals with osmosis. And osmosis is actually one of the more challenging topics for a lot of students in biology. Now, first of all, let me just recap what the question has stated so far. So 3 samples of blood were taken, right? And they were labeled as F, G, and H. Next, a solution containing salt was added onto all 3. Are we okay up to that point? All right. Now, the thing is, the salt solutions were of different concentrations, okay? So some of them may have contained more salt and others less so. Now, if you look at the table, you can clearly see this. So we have salt solution, percentage concentration added. So F had a percentage of 1, G had a concentration of 0.6, and H had the lowest salt concentration of 0.4. So we want to find out the effect that these different concentrations of salt solutions would have on the 3 samples of blood. Okay, so let's look at our table and the data provided. When it comes to F, at the end of the treatment, after 1 hour, the number of red blood cells remained the same. When it comes to G, there were fewer cells observed, meaning that some of the cells probably burst. I'm going to explain this later, okay? And then H, no cells observed. Whatever treatment that they underwent, caused the cells to burst, all of them, such that after an hour, no cells remained. Now, part A, name the physiological process being investigated in the experiment. This, of course, is going to be osmosis. Now, why do I say that this is osmosis? Because clearly we have movement of water molecules, and this only happens through the process of osmosis. Now, moving on to part B, account for the results made in blood sample F. Now, this question is asking us to explain, the state of the cells after the treatment with the salt solution. So first things first, I'm going to give just a brief review of osmosis. So osmosis is the process whereby water molecules will move from a region that is hypotonic to a region that is hypertonic, okay? And this happens across a semi-permeable membrane. Now, when you are comparing 2 solutions that are separated by a semi-permeable membrane, we use the following 3 terms: hypotonic, isotonic, and hypertonic. Essentially, these are just comparative terms, okay? So it's like saying person A is taller than person B. So automatically from this statement, you can deduce that person B is shorter than person A, right? Yeah. So hypertonic, isotonic, and hypotonic are also comparative terms. Now, if I were to say solution A is hypertonic to solution B, number 1, that means that solution A has a higher concentration of solutes than solution B, okay? And number 2, that also means that solution B is hypotonic to solution A. Bear in mind, these 2 solutions have been separated by a semi-permeable membrane. Now, when it comes to osmosis, water molecules will always move from the hypotonic solution to the hypertonic solution. Hypo to hyper, okay? Now, on to our last term, isotonic solutions. Now, these are 2 solutions that have the same concentration, equal concentration. And because of this, there is not going to be any net movement of water. So if you place a cell in an isotonic solution, it will remain the same after, okay? And that is the reason why when it comes to our body, you will find that a lot of the solutions that are present in our body, I'm talking about blood and so on, tend to be isotonic to our cells to ensure that, there's no net movement of water. Now, the reason is because if water molecules move into the cells, what will happen is that the cells will absorb water, swell, and burst, which, as you can imagine, is going to be fatal, deadly. Now, if it were the reverse, if our cells were placed in solutions that were hypertonic, what will happen is that they will lose water. They will lose water and they will shrink. Now, this cannot be allowed to happen because in organisms, the structure of a particular part of the body is always tied to its function. So if we have the cells in our body, shrinking, becoming wrinkled, it will definitely interfere with their functions. Now, in order to prevent either of these two scenarios, you're going to have our cells surrounded by isotonic solutions. Okay. Now, coming back to the table, if we look at F, the number remained the same. So what does this tell us about the salt solution that was added to F? It tells us that it was isotonic to the cytoplasm of the red blood cells. So the salt solution was isotonic to the red blood cells. And because of that, there was no net movement of water in and out of cells. So as a result, the cells were not hemolyzed. So what is hemolysis? Hemolysis is a process whereby animal cells will take in water by osmosis, become swollen, and finally burst. Okay? And that is what happened to the cells that are present in samples G and H. But in F, because the solution was isotonic, the cells remained the same. Part C. Account for the difference in appearance of red blood cells in samples F and G at the end of the experiment. Okay, let's look at our table first. If we look at G, okay, if we look at F, by the way, we have discussed F, right? We have talked about how the cells that were in F were exposed to an isotonic salt solution, and therefore we should expect at the end for them to retain their normal shape and size. Now, when it comes to sample G, what happened is that the number of red blood cells decreased, okay? Now, what happened is that some of these red blood cells absorbed too much water and ended up bursting, okay? They were hemolyzed, and that is why at the end, fewer cells remained. So that means that the salt solution was hypotonic to the cell cytoplasm, to the cytoplasm of red blood cells, okay? So, kama mnakumbuka, tulisema water molecules will always move from hypo to hyper. So, if the salt solution was hypotonic, that means water molecules moved from the salt solution and into the cytoplasm of the red blood cells. So, the red blood cells took in water by osmosis, became swollen, and some of them burst. And that is the reason why fewer red blood cells remained at the end of the treatment. Now, those that remained would be swollen, okay, because they have absorbed water by osmosis. On to part D. Now, what does part D say? With a reason, predict the possible observation that will be made if blood sample F was treated with a salt concentration of 2.6%. Okay, so 2.6% would be a hypertonic solution. Okay, so that means that this salt solution is going to be hypertonic to the cytoplasm of the red blood cells. So salt solution hypertonic, the red blood cells hypotonic. Water molecules therefore will move out of the red blood cells by osmosis and into the surrounding solutions. So what will happen to the cells? They will lose water and shrink. They are going to become crenated. But the number of red blood cells would remain the same. I mean, yes, they're going to be wrinkled, they are going to be crenated, but they're still going to be present, okay? They're still going to be there. So all the cells will still be there, only they will be crenated. Okay, moving on to the next question, part E. State the importance of the process being investigated during urine formation. So osmosis plays a huge role in the formation of urine. So where does this happen? In our kidneys, of course. Now, this involves reabsorption of water into the bloodstream. Moving on to our next question. A genetic investigation on a certain population established that in human beings, premature baldness is controlled by a dominant gene N, located on the Y chromosome. Part A. Work out the phenotypic ratio of the offspring of a couple comprising of a normal mother and a father who has premature baldness. Okay, so yeah. Now, before I tackle this question, let me briefly explain, because I know some of you are going to be Form 3 students currently, so I would like you to advance with me and understand this question. Now, in humans, we have two sex chromosomes, okay? We have the X chromosome and the Y chromosomes. Males have the genotype XY and females have the genotype XX, okay? Now, moving on to genes. The term gene was mentioned in the question. So before I explain about genes first, let me ask you this. Have you ever heard of DNA? I mean, who hasn't, right? So when it comes to DNA, deoxyribonucleic acid, this is a compound that contains genetic information. You know, it contains our genetic information. This is information that is passed from parents to offsprings. Now, genes, on the other hand, are specific segments of DNA, you know, specific parts of DNA. And these contain instructions that are responsible for the characteristics of an organism. So basically, genes determine the characteristics of an organism. And I'm talking about everything, okay, not everything, a lot about you. Your physical appearance in most cases, your intelligence, this is determined by genes in most cases, okay? They don't control all the traits, but a lot of them. Now, a single DNA molecule can contain thousands of genes. Thousands of genes. And these genes are going to be arranged in a specific order on the DNA molecule. Okay, now let me put all of this into context by giving you this example. I'm going to use a book. I know it's random, right? But your students, hopefully it will work. Imagine the DNA molecule as a book, okay? And the genes are the sentences within that book. So the sentences are arranged in a specific order to tell a certain story. So DNA, genes, okay? Now coming back to our question, we have a man and a woman with the following genotypes, okay? Now the question is focused on a specific trait, and that is premature baldness. Now, loss of hair is a normal part of aging, okay? But premature baldness is loss of hair that occurs at an earlier age than usual, okay? So I'm talking about people becoming bald or having thinning hair in their teens, you know, as adolescents, or in their 20s or even early 30s, you know, earlier than expected. And this is known as premature baldness. And this, of course, is genetic. It's determined by your genes. Now, this is controlled by a gene, and this gene is only present on the Y chromosomes, okay? So chromosomes are simply bundles of DNA. So you're going to have some genes present in both X and Y chromosomes. You're going to have others only present in the X chromosomes, and a few, such as this, are only present in the Y chromosome, right? So that means that this particular trait can only be present in males. Why? Because if you look at the two genotypes for females and males, only males have the Y chromosome. And if this gene is located on the Y chromosome, only males will have this particular trait. Now coming back to our question, our parental phenotypes are as such. The mother is normal. The father, of course, is bald. Our genotypes, mother XX, the father XY, but we are going to add N. Remember, we have been told to use capital N as representative of the gene. Now, this is only going to be present on the Y chromosome because this particular gene is absent on the X chromosome. So there we have it. Next step, we are going to separate these into their gametes. So XX and X1, just the way they are. Next step is fusion. We are going to cross the gametes, and this will lead us to the following 4 possibilities. Okay, so these are the possibilities of the genotypes of the F1 generation. Simply put that if, okay, they were to have an offspring, then there is a possibility of the following. Now, if you look at the genotypes, they are split. You have 2 females and 2 males. Because with every pregnancy, the possibility of having males and females is usually 50-50, okay? So in this case, with the daughters, they will always be normal. Because as stated, this gene is only present on the Y chromosome. So you will never have daughters that are prematurely bald. Now, some of you might be thinking, but we do have women that tend, to have thinning hair or have baldness and so on. Yes, this is a possibility. I'm just saying that it's not tied to this particular gene. It could be for a variety of other reasons. Now, when it comes to the males, what happens is that both the 2 males are going to be bald because this is present on the Y chromosome, and all boys always have a single Y chromosome, and the gene is also dominant, so it's going to be expressed when present. What do you mean by expressed? It's going to be visible in the physical characteristics, okay? It's going to be observable. So our phenotypic ratio, 2 normal daughters to 2 bald-headed sons. Part B. What is the probability that this couple would have a daughter with premature baldness? The possibility is 0, none. There's no possibility. Why? Because this gene is only present on the Y chromosomes. Daughters have a genotype of XX. So Y imetoka nowhere. Part C. Name 2 sex-linked traits in human beings associated with the X chromosomes. So I'm going to give the following: hemophilia and color blindness. On to question 4. During an ecological study in a lake ecosystem, a group of students made the following observations on the feeding relationships among some organisms. Okay, so we have lobsters that feed on algae, small fish feed on lobsters, worms, and caterpillars. Caterpillars feed on algae, worms feed on caterpillar, large fish feed on small fish, and then penguins feed on small fish, lobsters, and worms. Part A. From the students' observations, construct a food web for the ecosystem. Now, when it comes to food webs, what happens is that you need to have your connections ready. What do I mean by this? You need to, make the feeding relationships clear to yourself. Now, if you have a challenge in writing or in constructing food webs, then the easiest way for you to do so is you need to create food chains, okay? Now, for example, with the first one, lobsters feeding on algae, you are going to start with the producer. Always start with the producer. So in this case, in this ecosystem, the producer is the algae, okay? So in marine ecosystems, the largest producers are usually the algae. So you're going to have algae, and then you're going to draw an arrow away from the algae, connecting it to the lobster. Remember, the arrow is always pointing to where energy is being transferred to. So if the lobster feeds on the algae, that means that you're going to have transfer of energy in form of the food from algae to the lobsters. Now, once you have your food chains ready, it's simply a matter of connecting them. Part B. From the food web constructed in 4A, extract a food chain with the penguin as a secondary consumer. Okay, so this is whereby we are going to have algae, lobsters, and penguins. So that is the producer, that is the primary consumer, and that is the secondary consumer. Part C. Explain how spillage of oil from water vessels is likely to affect this lake ecosystem. So what will happen is that the layer of oil is going to coat the surface of the lake. This, of course, will prevent circulation of gases, specifically I'm talking about oxygen and carbon dioxide. So after some time, oxygen present in the water will be depleted, okay? This will cause the organisms to suffocate and ultimately die. Number 2, carbon dioxide concentration will also decrease. This will in turn interfere with the process of photosynthesis. Now, another effect is that the oil can end up clogging the respiratory surfaces of the aquatic organisms. This simply means that it can end up blocking respiratory surfaces such as gills, which of course will ultimately lead to the death of such animals. Question number 5. The following diagram represents part of a cell structure as seen under an electron microscope. A Roman 1. Identify the structure represented. This is the cell membrane. Roman 2. State two functions of the structure. Okay, so the cell membrane encloses the contents of the cell. It also allows for selective movement of materials in and out of the cell. Now, what do I mean by selective movement? So if you look at the diagram of the cell membrane, you can see that it has pores. These are small openings that are found on the cell membrane. Now, molecules that are small enough can easily pass across. Those that are larger cannot. So selective movement means that the cell membrane, selects what molecules can pass across simply based on their size. And of course, everything that is necessary and essential for the cell can easily pass across in and out of the cell. Roman number 3 suggests two ways in which the functioning of the structure can be impaired. So essentially the question is asking us, what can be done to the cell membrane that will interfere with its functioning? Prevent it from performing its function. Now in organisms, you know that the structure of any part of the body is usually tied to its function. So if you were to damage the structure, you will prevent it from functioning. So the cell membrane is made up of proteins, and proteins are sensitive to any changes in temperature and pH. So for example, if you have extreme temperatures, such as temperatures above 40 degrees Celsius, they will end up destroying the proteins. And this, of course, will interfere with the functioning of the cell membrane. Also, extreme pH. Strongly basic, strongly acidic. These will also damage the structure of the cell membrane by destroying the proteins. And in turn, you're going to have the cell membrane, not functioning properly or even ceasing to function at all. Part B. Name two structures found in plant cells but not in animal cells. Okay, so we have the sap vacuole, the cell wall, and chloroplasts. Although I would like to point out that when it comes to plant cells, not all plant cells have chloroplasts. A very good example are the epidermal cells. All epidermal cells lack chloroplasts except, of course, for the guard cells. Another example of plant cells that lack chloroplasts are the root hair cells. Part C. What is the purpose of staining cells before observing them under a light microscope? Now, this is to make the structures appear more distinct and clear. Moving on to section B of the paper. Question 6. An investigation was carried out to monitor trends of growth in a group of boys and girls for a period of 20 years. Their average weights were recorded at 2-year intervals as shown in the table. Okay, so we have the ages up to 20 years of age and the average weights in kilograms of the boys and girls. So if we look at those of boys, we are starting with 2.4, that is at birth, of course, moving on to 11, 14.5 until finally 48.6. So we are having an increase in weight as expected, of course. And then in the girls, same thing. But if you look closer, you're going to note that at certain ages, there is a difference in the weights of boys and girls who are at the same age. Okay, now we'll leave that discussion for later on. Part A. On the same axis, draw line graphs of average weights of girls and boys against their ages. So there we have it. Okay, let's tackle the questions related to the graph. Account for the trend in the growth of girls between the 14th and 18th year. Now, if we look at our table between the 14th and 18th year, 14th we have girls having an average of 35.4 kilograms, and 18 we have 52.6 kilograms. Okay, that's quite a fast rate of growth. Now, at this stage, this is where you have puberty. Okay, so you're going to have girls developing secondary sexual characteristics. I'm talking about widening of the hips, development of breasts, and so on. All of these are going to contribute to the increase in weight. Part D. Apart from sex and age, state 3 other factors that affect the rate of growth in humans. Now, we have BMR, the basal metabolic rate. We have one's health, okay? We have genes. Genes play a huge role in the rate of growth in humans. And also diet, the type of food that you eat. Part E. Other than weight, state a parameter that can be used to establish growth in humans. We can use height. F. Why do girls above 12 years require more iron in their diet than boys of the same age? Now, this is because on hitting puberty, girls menstruate, and through menstruation, they lose blood. So this is something that happens every cycle. So to compensate for the blood lost, they require more iron from their diet, from their food. Now, this is because iron is required for the production of hemoglobin. Okay, now we are left with the 2 essay questions, questions 7 and 8. So I'm going to provide you with the marking schemes as such. Ladies and gentlemen, brings us to the end of this amazing revision session.